Here you see lots of molecules moving randomly about due to their intrinsic kinetic energy. Now, in a cell, a phospholipid bilayer forms a boundary that keeps the environment inside the cell separate from the environment outside. So now, how will these molecules enter the cell? Welcome to BioWorld, where today we're going to answer that question. Let's have a look at our STPM syllabus. You need to know about passive transport, active transport, as well as a transport called cytosis. So come join me. Movement of molecules by passive transport is just like how a ball rolls down the hill from the higher gradient of the hill to the lower gradient of the hill. So when it comes to movement of molecules in biology, in passive transport, molecules move down the concentration gradient from a high concentration region to a low concentration region. As you already know, the plasma membrane keeps the internal environment of the cytoplasm separate from the external environment in the tissue fluid. To help explain the process of transport better, let me introduce some chemicals. These yellow ovals will represent lipid, while the red hexagons will represent either glucose or amino acids. The blue circles represent water, while the blue stars represent mineral ions. The first example of passive transport is simple diffusion. Simple diffusion is carried out by non-polar molecules. Therefore, polar molecules like glucose and amino acid, charged molecules like mineral ions, as well as bipolar molecules like water do not carry out simple diffusion. This process is limited to non-polar molecules such as lipid, fatty acid, glycerol, as well as vitamins A, D, E, and K. These molecules are lipid soluble. Therefore, they can readily diffuse through the phospholipid bilayer. The process of simple diffusion will come to a stop once equilibrium is achieved. This is when the concentration of the molecules outside the cell as well as inside the cell are equal. Another example of passive transport is osmosis. Osmosis is specific to water. Now, water is bipolar. However, it can cross the phospholipid bilayer because it is small. So, it is able to squeeze through the gaps between the phospholipid molecules to reach equilibrium. In osmosis, when the water molecules are equal outside as well as inside the cell, we say the cell has become isoosmotic. The third example of passive transport is facilitated diffusion. Facilitated diffusion is done by both polar as well as charged molecules. So, the non-polar molecule lipid will not carry out facilitated diffusion. Now, the word facilitated means helped. So, to carry out this transport, proteins are needed to help. The proteins are intrinsic proteins such as channel proteins as well as carrier proteins. The molecules will choose which protein to use for transport based on their molecular size. So the smaller molecules such as mineral ions will travel through the channel proteins down the concentration gradient. Earlier, we discussed how water by osmosis can squeeze through the spaces between the phospholipid molecules. But we find water can also carry out a form of facilitated diffusion by using channel proteins called aquaporins. Aqua meaning water, por referring to another name of channel protein, that is pore protein, and the in at the end refers to protein. So water molecules, especially in the kidney, need to be transported very quickly, so they will diffuse through the aquaporin molecules by facilitated diffusion. But since the movement is down the water potential gradient, therefore it is still considered a form of osmosis. Larger molecules such as glucose and amino acids cannot slip through the pore of the channel proteins. So they require the use of carrier proteins. The carrier proteins can rearrange themselves so that these large molecules can enter through the proteins down the concentration gradient. But all these processes also come to a stop once equilibrium is achieved, that is, once all the molecules are equal on both sides of the cell. We'll move on to active transport. Now, just like how a ball cannot go uphill unless we give it a kick, molecules in biology too cannot move against the concentration gradient from low concentration region to high concentration region unless it is provided with a kick in the form of ATP. Active transport is carried out by lipids, glucose, amino acids, as well as mineral ions, where they are transported against the concentration gradient. However, water molecules, since they are transported by osmosis, cannot do active transport. Active transport uses proteins, specifically carrier proteins. Therefore, channel proteins are not necessary. Besides that, active transport, of course, requires ATP. So as you can see now, the glucose or amino acid molecules, the lipid molecules, as well as the mineral ions presently are in equilibrium. So the process of passive transport would have come to a stop. But let's say the cell needs more lipid, more glucose, more amino acids, as well as more mineral ions. So that is when the ATP will change the conformation of the carrier protein so that it now transports molecules from a low concentration region to a high concentration region. Now this process will only stop once accumulation is complete. Now there are no more molecules to be transported from outside the cell to inside the cell. Another instance where active transport will come to a stop is when the cell runs out of ATP. Transport of molecule across the plasma membrane can be represented by a graph. Here, the y-axis shows us the rate of molecule uptake by the cell, and the x-axis shows us the concentration of the molecule. Let's look at the green graph. In this graph, we can see that as we increase the concentration of the molecule, the cell is able to increase its rate of transport. This is seen in transports like simple diffusion involving lipids and osmosis involving water. The reason why both these methods of transport can be carried out so efficiently is because they cross using the phospholipid bilayer, which is abundant in the cell. In contrast, the red line shows us that at a certain point, when we increase the concentration of the molecule, the rate of transport of the molecule does not increase. Instead, it remains constant. We see this pattern in transports such as facilitated diffusion and active transport. These transports involve the transport of molecules such as glucose, amino acid, or even mineral ions. Now, the reason why increase in molecule is not complemented by increase in transport is because the proteins, either carrier proteins or channel protein in facilitated diffusion, behave as limiting factors. What we mean by limiting factor is that these proteins may be involved in transporting. So, when we increase the concentration of the molecules, these molecules have to wait their turn. So that is why, although the molecules are being increased, the transport rate is not increasing. Now I'm going to talk about a transport called cytosis, which happens across the plasma membrane. To start off, I'll talk about endocytosis, that is the transport of molecules from outside the cell into the cell. There are two types of endocytosis. The first one called phagocytosis, involving bulk materials such as carbohydrates and protein. The other type is known as pinocytosis, which involves fluid droplets. Now, what happens during endocytosis is that the plasma membrane will infold. So, when it infolds, it forms a pocket. The bulk material or the fluid droplets will move into that pocket, and the plasma membrane then rearranges itself so that the bulk material and the fluid droplets are trapped in vacuoles or vesicles. Exocytosis, on the other hand, is the transport of molecules from inside the cell to outside the cell. This occurs when an organelle, like Golgi apparatus, begins to synthesize secretory vesicles. Let me magnify the secretory vesicle. Now, inside the secretory vesicle, there can be hydrolytic enzymes, secretions, or mucus. So, this secretory vesicle then will move towards the plasma membrane. At the plasma membrane, the membrane of the vesicle and the plasma membrane will fuse so that the secretion can be released to outside the cell. Then the plasma membrane will rearrange itself so that the membrane of the secretory vesicle fuses with the plasma membrane. That we now understand how molecules travel. Bye bye. See you next time.